Okay, great. Thank you, Marcus. So let me get my screen up here. So I think you can see that now. Yes?
Yes.
Okay, perfect. So thank you for that really kind introduction, Marcus. I really appreciate it, and thank you for asking me to speak to you today about AI and biotechnology. So I'm going to focus my talk about a recent study by the U.S. National Academies of Sciences, Engineering, and Medicine that I co-chaired, in which we were asked to look at this topic. And you'll see that I'm going to touch on some of the same points that Marcus just spoke about in his presentation. So the report was authored by the Committee on Assessing and Navigating Biosecurity Concerns and Benefits of AI Use in the Life Sciences. And I'd like to thank the U.S. Department of Defense, which sponsored the study. And before I dive into the report, though, I want to just give you a brief introduction on how the Academy's process works, just so you can get a sense for, how these reports come to be. And so the National Academies of Sciences and Art was established in 1863 by an act of Congress in the middle of the Civil War, when President Abraham Lincoln and others were facing some unprecedented scientific and technical challenges and needed advice that was free from partisan politics. And so during its first decades of existence, a lot of the questions that the government asked of the academies were military related. But President Lincoln founded the academies with a mission that went beyond the science and technology of warfighting. And so a century later, the National Academy of Engineering was discovered, followed by the Institute of Medicine, which is now called the National Academy of Medicine. So there are the three national academies. And really the most recognizable products of the academies are these so-called consensus studies reports. And these are really considered by the government, academia, and the general public as the gold standard for objective science-based advice. And so there are 5 stages in the consensus study process. The first is the development and initiation of the study, which usually comes from a request from either a government agency or sometimes a foundation. There's then formation of the committee. So there'll be a call for nominations. The staff reviews the nominations and tries to find a good balance of expertise, backgrounds, and other elements of diversity with respect to the members of the study committee. The academies ensure that any biases are balanced, and committee members are free of any conflict of interest. Then the committee gets together, gathers information, does its analysis, and writes a report. That report undergoes expert review, and then we do outreach, which is what I'm doing today. So that's just a little bit of an overview of the process here. So the other thing I want to do before I start is just to thank a number of people. So I have to acknowledge the amazing members of the study committee, with special thanks to our co-chair, Linda Stewart. We're fortunate to have an outstanding committee of 12 experts from academia, industry, and government with varied expertise: virology, AI, biosecurity, synthetic biology, computer science. So it was really a great group to work with over the months that we did this study. And then I also have to thank the Academy staff. So this report fell under the scope of 3 different scientific boards in the academies, and the study was fast-tracked, which meant that the staff needed to be extra diligent to keep us on track, support us, and assist with writing the report. Okay, so let me begin by the statement of tasks. So this is what was given to us from the sponsor, what they asked us to do, and it's shown here. And the central purposes of the report are twofold. Number one, to provide an assessment of how AI, and more specifically, AI-enabled biological tools trained on biological data and intended for beneficial applications, may be misused for harmful applications, particularly by facilitating the design of transmissible biological agents that could pose epidemic and pandemic scale threats. And so we were really asked to focus on these transmissible agents. And then secondly, the question was, how can AI be used to mitigate those risks? So what are the beneficial applications of AI that can be employed here to try to either prevent or avoid those kinds of risks, and then potentially to respond should something bad happen. And I want to just note that general purpose AI models such as ChatGPT were deemed to be outside the scope of the study. So we really wanted to focus on AI-enabled biological tools. Okay, so the committee's approach was to host a series of information gathering sessions to hear from experts across the public and the private sector, including the leading developers of AI tools. The sessions covered topics such as applications of generative AI tools for biology, biological data security implications, and high-performance computing. The report underwent rigorous peer review by 9 experts whose expertise spanned across various disciplines, including computer science, molecular biology, and national security. And we thank them because those reviews allowed us to improve and enhance the report. The committee also references and builds on a report that was previously published by the National Academies that Marcus mentioned, called Biodefense in the Age of Synthetic Biology, which I also had the privilege of chairing, and which provides a risk assessment framework for synthetic biology capabilities. The new report, with its focus on AI, represents the outcome of a review of the capabilities of AI-enabled biological tools by the committee, and we think that it can be used together with this previous framework for a full consideration of the threat landscape and risk assessment. And I would note that when we did this report in 2018, AI was barely on the radar screen. And it's just an indication of how very rapidly this field has moved. And I would also point out that all of these reports are available for free at the National Academy's website. So the report's organized into 5 main chapters that are shown here, in addition to an executive summary and appendices. So chapter 1 provides an introduction to the convergence of the AI and the life sciences and outlines the committee's approach to addressing the statement of task. In chapter 2, we talk about the capabilities of AI-enabled biological tools in synthetic biology applications, and in particular, the design, build, test, learn process. Chapter 3 explores how current AI biological tools can uniquely enable biological design when the tasks involve different levels of complexity, and in turn addresses the implications with respect to biosecurity risks. Chapter 4 covers the ways AI can be leveraged to mitigate risks in bolstering biosurveillance capability and in accelerating the development of medical countermeasures in response to biological threats. And Chapter 5 discusses the importance of biological data sets that can be used to train AI and drive innovation. And I'll mention right here, someone asked a question in the chat about poisoning data sets and that sort of thing. That's all there in Chapter 5, although I'm not going to really talk about that in any detail in this presentation. And then Appendix A is a really nice appendix that gives an in-depth review of current AI biological models and their applications, limitations, and available training data. And we thank Brian He at Stanford University for writing that appendix for us. So let me start off by talking about the convergence of AI and life sciences. And Marcus already talked about this a little bit, but I'll just repeat it here. The interest in AI broadly and its integration in our lives in various ways is widespread, and this includes the life sciences. Now, computational methods in the life sciences isn't new per se, but the increasing availability of data, along with advances in technologies to generate more and more data and computing power, have contributed to this convergence of AI in the life sciences. The impact of AI to accelerate and enhance research, discovery, design, predictive power, and understanding biological systems has tremendous promise, with applications in drug discovery, precision health, interpreting medical imaging, optimizing bioengineering, just to name a few. So one of the major breakthroughs was achieved by the release of AlphaFold and RosettaFold for protein structure prediction. And we know that last year's Nobel Prize in Chemistry was awarded to the researchers working on computational protein design and structure prediction. As with other new biotechnologies in the past, the question of whether the technology can be exploited for harmful applications has been a topic of debate. And Marcus talked about this. And in particular, the question is, can the application of AI lead to the creation and deployment of a biological threat? This concern is, of course, not new, but how AI may help facilitate this risk is. And so I like to think about this as sort of the convergence, in a way, of two dual-use technologies. And so the conversations regarding the risks specific to AI involve the notion that AI may lower some of the barriers required to cause harm, such as expertise and resources, as was previously noted. And so the committee considered that the core of the question we were asked to address is how AI biological tools can uniquely enable this risk, right? So we know someone can synthesize the DNA for a pathogen, right? But the question is, can AI enhance this ability, right? So what is the capability uplift? I know there's been some objections to the use of the term uplift, but that's what one of the terms we used in the report. And we also refer to this as the Delta AI. So first, I want to just go through a quick primer on different AI biological models to frame how we considered the current capabilities and limitations and what they meant when we asked questions about different applications. So first, our foundational models trained on large-scale biological data sets using unsupervised objectives, and these learn to capture the underlying distributions of biological data. These models, which are exemplified by protein language models like ESM and genomic language models like EVO, demonstrate remarkable generalist capabilities. There are then generative models, which are often built upon or closely related to foundation models, that enable efficient sampling from learned biological distributions and have shown particular promise in generating new protein sequences and structures. Predictive models, on the other hand, focus on mapping between different biological modalities, such as predicting protein structure from sequence, for example, AlphaFold, or inferring epigenomic features from DNA sequence. So Informer is an example of this. And then there are design models which leverage both generative and predictive capabilities to produce biological entities with desired properties, as seen in protein engineering and promoter design tasks. And as you'll see, this is really where the committee focused much of our attention. And then finally, again, I just want to mention that there's a lot more detail about these different types of models in that appendix that I talked about if you're more interested in reading up on that. So I'm not going to go through the details of this slide, but this is just some examples of AI biological models that were developed or released in the span of 3 years preceding the study, and they're grouped according to broad categories. And just in the past few months, we've seen a few notable models published even since the completion of the report. So again, this is just a very rapidly advancing field. And so now let's take a closer look at the capabilities and impact of AI-enabled biological tools in synthetic biology. So the first question is, what is synthetic biology? And there have been many different, attempts to define synthetic biology. Our committee used the definition from the 2018 report that I mentioned earlier, which defines synthetic biology as concepts, approaches, and tools that enable the modification or creation of biological organisms. And so one distillation of this was that synthetic biology aims to improve the process of bioengineering. And the approach to that end is the design, build, test, learn process. So as you know, DBTL is an iterative process for designing, constructing, and evaluating biological systems with a built-in feedback loop for refining designs based on experimental data. And so the question is, do any AI-enabled biological tools enhance this process? Or again, what's the delta AI? And so this diagram just illustrates this process going from ideas to design to build, test, learn. And you'll see this little gray triangle at the top, which kind of gives away the punchline to some degree, which is that the delta AI gets lower and lower as you move from left to right in this process. So an emerging area of research is also the use of AI-enabled biological tools in biosecurity applications that can help mitigate biological threats. For example, AI can help enhance biosurveillance in monitoring and early detection of outbreaks, as well as accelerate the design of medical countermeasures. The ability to respond rapidly in the case of public health events is an important measure in limiting the scale and scope of harmful consequences. And I also want to mention here that, we really focused on human health here, but, we also have to be very concerned about animal health, plant health, because this is a much broader topic. And I think that the recommendations we made here also apply more broadly. And aside from the role in response to a biological threat, AI can also be used to detect potential misuse, such as in DNA synthesis screening, as Marcus talked about a few minutes ago. However, this is a relatively new area of research. And so keeping the design, build, test, learn cycle in mind, we considered what the implications are for increasing potential security risks, or again, how AI-enabled biological tools uniquely enable harmful applications. And so to break this down, let's look at 3 different biological problems that represent the biosecurity risk or threat. And those 3 problems are design of biomolecules, such as toxins, modifying an existing pathogen to make it more dangerous, or designing a novel bacterium or virus. Okay, and we really focused on viruses because we didn't have a lot of time to get this study done. And so these 3 problems or tasks differ in how complex the task is, and correspondingly, the scale of potential impact. And so this figure just aims to show that going from top to bottom, we have design of a molecule, modification of a microbe, and then design of viruses. And on the x-axis here, just shows some of the considerations going from the top. There are very few considerations and they're rather simple. As we go down, there are more and more consideration, more steps in order to get to the output here. And so you can see, and I'll talk about this in a little bit more detail, molecular design has been solved. As we know, there's already been a Nobel Prize for that. And also as we go from top to bottom, there's increasing technical difficulty, but also increase in the potential level of impact should something weaponized. And so if we look at current AI-enabled biological tools, they're capable of various modeling tasks, such as design and prediction, when it comes to single biomolecules, such as a protein. This capability can be exploited to design a novel toxin or redesign a known toxin with different amino acid building blocks to potentially bypass simple homology-based DNA screening. However, we would note that the potential scale of impact in the context of misuse here is usually limited because these molecules are not transmissible. Now let's consider the problem of modifying an existing transmissible biological agent or designing a new one. On the surface, the task of modifying a single or even a combination of traits that play a role in virulence may seem analogous to designing or modifying a biomolecule. And there are certainly current tools that can predict some phenotypes from genotypes. However, we have to remember that biological systems such as viruses are complex. and phenotypes result from intricate interactions at the molecular level, environmental factors, host pathogen interactions, and they're always under evolutionary processes that can be difficult to model accurately. Moreover, phenotypes can change over time due to selection, requiring dynamic modeling that current models and tools are not yet capable of doing with great accuracy. The ability to design a completely novel, self-replicating organism is faced with the same problems of a highly complicated task in whole systems modeling, and there are currently no useful data sets or AI tools with this capability. So designing something new at the present time would be incredibly difficult. In both of these latter two scenarios, modification or novel design, current AI biological tools face the same limitations. The lack of sufficient data sets limits capabilities in modeling and predicting the biological complexity. Importantly, AI-enabled biological tools do not reduce the barriers and bottlenecks in the physical production or the build test phase of the cycle. However, future advances in automated laboratories may improve efficiency and throughput in a so-called lab in a loop, right, where you get AI-driven experimental design and automation of the build and test work. However, current automated labs are typically application-specific and resource-intensive, and they require a large amount of human input and capital investment. The report also considered future advances in AI that may be a significant increase in capability, such as advances with LLMs and foundation models, and the use of synthetic data, so artificial data that are generated to mimic real-world data, would also lower the requirement of generating large and varied types of experimental data sets that are resource intensive. And so this figure just summarizes what I just discussed in terms of looking at the different capabilities and limitations of AI tools in biological design tasks. So at the bottom, again, there's the design of an individual molecules. And we know now that this could be done. So the AI could really help with ideation and design to build a single molecule, right, to clone it into a bacterium or whatever you want to do, and produce that protein is relatively simple. And so this is really a concern that's real in today's environment. However, to go up to the top here and think about self-replicating organisms, first of all, the digital side of this is much more difficult because it's a systems biology problem. And then you have to be able to go from that digital design to produce an actual physical entity. And even with an existing known virus, for example, it's not always easy to go from having a genome sequence to actually producing that virus. So the barriers there are much more significant. And so this brings us to the question of how to mitigate these potential risks and not hinder innovation in AI and the life sciences. So the first consideration is that AI can be leveraged to address these risks, and I'll discuss how shortly. Of course, advances in these applications need additional and continued support for research and development. Secondly, AI relies on data for training, and developing the appropriate infrastructure and resources to manage the data ecosystem and lifecycle will be important to driving AI innovation. And finally, the report recommends a strategy for how to consider the risks that takes into account all the factors at play at this intersection of AI and the life sciences. And so now I want to take you through a slightly deeper discussion of our recommendations. And these recommendations were made to the US government since it sponsored the report, but I think they really apply more broadly to the broader global community because they get to some of the things that we've already been talking about this afternoon. So We recommend that federal agencies funding research continue to invest in research that advance and drive AI innovations in the life sciences. I mean, this ought to be obvious, right? But we felt it was really important to restate that very clearly here. If we want to maximize the benefits, we need to support the research. We also recommended that a public-private partnership be established to enable rapid activation and access to AI resources and tools in response to a public health emergency, similar to Operation Warp Speed that was used here in the US to develop COVID-19 vaccines, and also the COVID-19 High Performance Computing Consortium, which were really beneficial during the pandemic. The committee noted that in light of the rapid pace of development of AI-enabled biological tools and the continued collection of training data that could lead to newer improved capabilities of such models, it's difficult to predict the future. Therefore, the committee recommended the adoption of an if-then approach to guide future vulnerability and risk assessment and mitigation. The if-then strategy accounts for these dynamics without being prescriptive or restrictive about predicting future capabilities based on current knowledge and capabilities, because that knowledge and capabilities are rapidly changing. An if-then approach would allow for the continual assessment and periodic reassessment of vulnerabilities and risks using a principles-based thought framework. These evaluations should be anchored in both data availability and the associated emerging AI capabilities, as well as real-world threat models, many of which were articulated in the 2018 report. In addition, the if-then strategy should establish benchmarks and metrics to track data availability and associated capability progress, as well as a set of thresholds that, once surpassed by emerging capabilities, activate risk assessment and mitigation. So, for example, I already mentioned that right now there are not really good tools for mapping phenotypes to genotypes, right? But as those tools become more developed, then we have to think about what the potential risks are that are associated with those advancements. The committee also considered a number of steps to maximize the benefits of utilizing AI to drive advances in AI capabilities. The generation, curation, and preservation of a data repository requires significant long-term investments in laboratory and computing infrastructure capacity, as well as subject matter expertise. Building new data resources and the strategic collection of AI-ready biological data sets should be a research priority to maintain scientific competitiveness and innovation, and again, to ensure that we can really reap the benefits before there are any potential misuse of these resources. And so in summary, the application of AI in the life sciences is a very dynamic area of development. The capability uplift of current AI biological tools is most significant for design and much less so in the build and test areas of the workflow. Capabilities are limited by the biological complexity of many of the tasks that we're thinking about, the availability of biological data for training models, and the need for experimental validation. High-quality biological data are crucial for training models and for the innovation associated with those models, and then maximizing beneficial applications in biosecurity against the potential risks can be achieved by investing in research and infrastructure and in conjunction with ongoing risk assessment. And I just want to finish up with a sort of a personal note, which is that, I'm really concerned that the public trust in science is eroding globally, and especially so in the US, where the current administration is actively encouraging people not to trust science. And so I think that we, the scientific community, really have to pay attention to the advances that we are making and in this field of AI and biotech, to ensure that we are using these technologies safely, and that, as Marcus noted and as Michael noted, we have to show that we're really paying attention to the potential risks, and showing that we are being responsible. And so I would stop there, and I'll be happy to answer any questions. Thank you.